It's a great privilege for me to give this talk, and I'm really grateful for the kind invitation. It's a pity that we can't do this in person, but I hope that every one of us takes advantage of all the benefits that a virtual platform offers. The purpose of this talk is to give you a flavor of this fascinating field known as biophysics. The field is so vast that I can't even touch on the large majority of aspects, but I hope I will do justice with a few examples that I've selected in this talk. So this will be merely a quick bird's eye view of biophysics, with a particular emphasis on molecular biophysics. Let's start with the basic question, what is biophysics? This field is often thought of as some mix between biology and physics. For example, you mix 40% biology and 60% physics for one type of problem, and a different mixture for another type of problem. But this is wrong. You're not half a biologist and half a physicist when working in this field. Say we want to study some biological system. The biophysicist takes the lens of physics, the principles and methods of physics, to gain a deep understanding of the biological system of interest. And this can literally be any component of any biological system. There are two approaches to this field of research that I'd recommend. Firstly, the application of physics to a biological context. That is, think like a physicist while looking at biology. We need to look at all of nature through the eyes of a physicist. Secondly, be problem-oriented. That is, use the best available tools to investigate a particular question at a deep quantitative level. And this will most certainly include sophisticated tools developed by physicists. In fact, we are living in a very privileged era where we have access to stunningly sophisticated computational and experimental tools to address basically any complex problem in nature at a remarkable level of detail. And the complex problems of our day demand multidisciplinary approaches. Let me give you at this stage two simple examples of how one can look at biology through the eyes of a physicist. Let's first consider dynamics at the level of a biological population. Have you ever wondered why fish often swim in schools and birds fly in V-shaped formations? It's to save energy. It's literally built into their DNA to flock together in order to be more energy efficient. We can, for example, simulate this behavior entirely using computational fluid dynamics and then correlate our simulations with experimental data and gain an even deeper understanding when manipulating these patterns by adding chemicals or other perturbations. The next example is at the level of a single organism, an insect walking on water. This is actually a physics problem. The insect makes use of special properties of water, in this case the surface tension, to balance its weight. Let me take this a step further and give three motivations for why biophysics research is very important. Firstly, physics brings the understanding of biology to a deeper level. The core questions of our day may come from one discipline, but the solutions are often found from integration with other disciplines. And the integration of physics to resolve questions in biology is a beautiful example of this because of the nature of physics, the tools it offers to give quantifiable descriptions, to develop mathematical models, to make reliable predictions, and so on. The story goes that while administering a second year exam at Princeton University, Einstein's teaching assistant asked him, Dr. Einstein, aren't these the same questions as last year's final exam? Einstein famously responded, Yes, but this year the answers are different. This may be quite funny, but it is a pretty insightful answer considering the development of science, and it is particularly true for 21st century biology, especially when it comes to biological molecules. Most of the experimental and computational tools used in frontier science have been developed by physicists, and the developments are ongoing. So we are continuously getting deeper answers to some questions. In fact, it's hard to think about modern day molecular biology without physics. At Friday's Biophysics Winter School, Dr. Martin Frieder from the World Health Organization gave a brilliant talk, and he said in his talk that you will not be able to successfully develop vaccines without biophysics. He mentioned that there's currently a vaccine company hiring biophysicists because they know that the next generation vaccines cannot be developed without the involvement of biophysics. Even nature physics has an eye on biophysics, especially where we can learn more about physics by studying biological processes. As highlighted in this editorial, biophysics is firmly part of the remit of nature physics, and many biologists are appealing to physicists to help them make sense of their data. The second motivation for biophysics that I'd like to offer is that biology offers a testing ground for some theories of physics, in particular for non-equilibrium statistical physics, as highlighted in this commentary. The author of this commentary, Christopher Jarzynski, is of course known for fluctuation theorems, for which small biological systems are excellent testing grounds. Some of this is discussed in my physics reports paper published earlier this year. In this review, we considered a broad range of thermodynamic processes, not only for biological systems, but ranging from various types of quantum systems to stellar galaxies and cosmological models. The third motivation I'd like to give is that biophysics is very important for innovations. The term century of biology was coined in the context of technological developments, and there's quite a long list of critical global challenges where biophysics plays an indispensable role. Here are some of the major ones that I've listed. I don't want to spend too much time on this now, but let's only have a brief look at a few examples from quantum biology. So it's very interesting to realize that many of the key enabling technologies that the EU is seeking to develop are based on quantum mechanical phenomena and find examples in nature. In other words, mankind is desperately seeking technological solutions, some of which already exist in nature. Here are a few examples of what life can do that we cannot. I've shown examples from photonics, nanotechnology, nanoelectronics, advanced materials, and PV, with examples of biological systems that can do what we like to do in some of our own technologies. One of the fascinating aspects of biology is that besides the enormous diversity in organisms and processes, we're also dealing with many scales in both space and time. A plant like a tree is the system with the largest range of time and length scales, 12 orders of magnitude along the spatial scale, and a staggering 21 orders of magnitude along the time scale, from the absorption of a photon within 1 femtosecond, 10 to the minus 15 seconds, to seasonal variations of the tree. This is pretty amazing. Another fascinating aspect is that of emergence. It's a reality that new properties or functions emerge at a higher level of organization, which you cannot predict from a lower level. And in this way, you get a living system which is ultimately guided by molecular processes. And that's really mind-boggling if you think about this. The last aspect I'd like to highlight here is that living systems solve physics problems at all levels of organization, all levels of complexity. And let me illustrate this at the level of the cell. Shown here are the three main types of biological cells, classified into bacterial cells, plant cells, and animal cells. And this is a typical picture of a plant cell, which you'll find in elementary biology books. So the question here, when looking at a system like this, is how should a physicist look at this? Firstly, appreciate that cells are amazing factories. Actually, they are much more than microscaled factories. They function as mini cities, with one unit functioning as an energy powerhouse, another as a garbage company, another as a post office, and so on. Let's now use some mathematical tools to construct a very simple model of biological cell. And I'm going to use the concept of a minimal cell, which describes the minimum number of biomolecules, reaction pathways, and compartments necessary and sufficient for proper cellular function. I'll go over this very quickly just to give you a flavor of it. Let's assume circular symmetry and only the radial component X. In this diagram, each arrow represents a reaction between two molecules. The simplest process is described by a diffusion equation, for example, diffusion of small molecules across a membrane. So here's our diffusion equation. But even defining this simple process is very challenging. There are, for example, boundary conditions which are often ill-defined. And the molecules undergo chemical reactions with other molecules, so there must be source and sink terms to this equation. Also, since the rates of various reactions in here must match, there are feedback processes, which means that the rates are interdependent. So even describing this simple process, the simplest of processes here, requires us to solve a set of coupled partial differential equations, PDEs. In this diagram, which is the simplest version of a possibly functioning cell, all the components are interconnected. So counting all these arrows gives us then 30 coupled PDEs. So there's no way that Mathematica can help you to solve this on a laptop. Even a big HPC cluster will take very long to do this reaction, this calculation for you. And a real cell is incredibly more complex. Here's a diagram of metabolic pathways in one of our cells. And you can just see the staggering complexity of this. So, how many coupled PDEs do we have to solve now? So I hope that by this example that you can get an idea of how extremely complex a cell is. Let's just see, how should I close this? Okay, there we go. Okay, let's now zoom into a plant cell. So here we have a light micrograph of a plant cell where you can see that they are packed with chloroplasts, these green balls inside. And each of these chloroplasts consists of folded membranes. And inside such a stack of membranes are all the macromolecular structures that are responsible for the light-dependent processes of photosynthesis. So here you can see a photosystem, and one small part of such a photosystem is a protein which binds a bag of pigments, which are the light-dependent or light-absorbing molecules. These proteins are amazing systems. They are, in fact, molecular machines, the molecular machines that are responsible for virtually every function in the cell. Due to their extreme importance, they are sometimes called the hydrogen atom of life. As a physicist, what I find extremely interesting about proteins is that they're situated on the border between the classical and the quantum regimes. Even though this is a rather fuzzy, ill-defined border, proteins make up very interesting objects for studies of quantum biology, where we want to know how non-trivial quantum effects may be relevant for biological function. And this relation between the classical and the quantum worlds in a biological context is exactly what quantum biology is. When there are some biological features and functions that cannot be accurately be described by the classical laws of physics, then we're dealing with quantum biology. A science journalist, Philip Ball, has made an interesting comment that the key to practical quantum computing and high efficiency solar cells may lie in the messy green world outside the physics lab. That's a pretty bold statement. But why is he referring to solar cells? Well, that's because photosynthetic organisms contain highly efficient, highly adaptable solar cells called photosystems, which are part of a very complex photosynthetic machinery to convert solar energy into useful forms of energy. And why did he refer to quantum computing? Because it seems that some biological systems have the ability to sustain quantum coherences for times as long as a few picoseconds at room temperature, despite their noisy environment. I would highly recommend this headline review article, which reflects a little bit on the history of quantum biology, but much more on the current state of the art and the predicted future. Let me now show you one example of a clear demonstration of quantum biology. For this, we consider the main light harvesting complex of plants. It consists of three identical protein subunits and 54 pigments, which are mainly chlorophyll molecules. These pigments are all packed into the size of a disc of a diameter of about 7 nanometers and a thickness of 4 nanometers. And this is a pretty high density. This density gives rise to so-called molecular excitons, which are excitations that are coherently shared by a few molecules due to the overlap of their wave functions. The physiological benefits of this phenomenon are huge. Molecular excitons give rise to, firstly, more efficient light absorption because of an increased absorption cross-section. And secondly, they lead to faster energy funneling, which is faster population of low energy states. Because in a quantum system, coupling with vibrations concentrates an excitation in a low energy state. This is in contrast to a classical thermostat for which equipartition applies and therefore all states have the same probability. Then there's also faster energy transfer taking place between different pigments and much less energy trapping, which may otherwise lead to a loss of the exciton through recombination or thermal decay. These last two aspects can be explained by thinking what would happen if you try to play soccer with a golf ball on a grass field. Why is that not going to work? It's because of the roughness of the landscape. The roughness of the landscape is of the same order of magnitude as the dimensions of the ball, which means that the ball is going to get stuck. It's going to get trapped every few meters. But a soccer ball is big enough to go smoothly over the landscape. The same applies to excitations in an energy landscape. If your excitation is physically big, it will not get stuck in an energy trap while diffusing in the light harvesting antenna network. One of my PhD students spoke yesterday in the theoretical and computational physics track about some of the theoretical aspects of light harvesting complexes. It's not so trivial to accurately calculate linear spectra of light harvesting complexes and to determine their Hamiltonians. If you have missed the talk, there's an archive paper that we have published a couple of weeks ago, and you can look at this paper which describes part of this work. Let me now show you one example of an experimental study on excitonic dynamics. The biological context is that diatom, and diatoms are amazing organisms. They are a major group of algae and mostly unicellular with a size of between 1 and 100 microns. Here you see many different shapes of their outer shell, which is made of silica. If you think about this, it's amazing what genes can make with silica. So I took the major light harvesting complex of a particular type of diatom, and by doing that, I discovered a new light harvesting state indicated by the red population here. We then performed excitonic modeling based on an open quantum systems approach to calculate the coupling between these three key chlorophyll molecules that give rise to this new redshifted state. And very interestingly, this new state corresponds to reduced excitonic coupling between these three chlorophyll molecules. But this reduced coupling gives rise to an enhanced light harvesting efficiency, which is completely counterintuitive, because when the excitonic coupling decreases, the system becomes more susceptible to disorder, noise in the system, and one would therefore expect a reduction in the light harvesting efficiency, but the opposite effect was found. In the remaining time, I would like to show a few selected examples of experimental methods to investigate individual biomolecules. The first is the fascinating technology known as DNA origami. Since DNA is fully programmable, you can design almost any two-dimensional or three-dimensional nanostructure using a single strand of DNA. DNA origami is commonly used as molecular breadboards onto which proteins and other macromolecules can bind at targeted positions with nanometer precision. Here at the bottom is an example where short pieces of DNA were used as staples or as a glue to fold a long piece of viral genome into a smiley face. In this second example, this light scattering was used to determine the weight of a protein, biological molecules. So, and this was shown a few years ago that this scattering can be used to determine the mass or the size of a particle as small as a protein. It's actually not the scattered light alone that was used, because that's too weak, but the interference of the scattering with the light that is back reflected from a nearby interface was used. In my lab, we are currently developing a technique where we are using the same interference between the scattering and the back reflection as a source of photons to track individual particles like a protein in real time. And I'd like to highlight this review paper where we are discussing real-time single particle tracking in detail. This is a relatively new but hugely underdeveloped technique with the potential to revolutionize single molecule experiments. So this brings me to single molecule spectroscopy, one of the experimental techniques that I'm specializing in. It's an extremely sensitive and selective method and allows one to study the behavior of a single molecule of protein in real time. There are smart ways to retrieve information about protein dynamics and photophysical processes down to ultra-short timescales, on the order of a few femtoseconds or tens of femtoseconds, on single molecules or proteins. Many of these phenomena are often studied in parallel, giving multidimensional information about the molecular systems and their local environments. The key here is to extract as much information as possible from each detected photon, and for this purpose it's useful to use interferometry for ultra-fast single molecule spectroscopy, or time tagging the photons, modulating the intensity, doing first order or second order photon correlation studies, or resolving the polarization or the spectra, the full spectra, and then to correlate all these parameters and their changes as a function of time. Here is an example of spectral diffusion, the time-dependent dynamics of the emission spectrum of one of the subunits of plant photosystem 1. In this example, I discovered that the subunits of photosystem 1 can reversibly switch off their red emission. So as shown in this example. So, and this happens due to a change as small as zero point 0.7 to zero point 0.8 angstroms in the separation between two key chlorophyll molecules, chlorophyll A5 and B5. It's really amazing that such a small physical change can drastically change the biological function of the light harvesting unit. Here is an example of how we used light to control the interaction between two individual proteins. One very small protein called the orange carotenoid protein, it's a small protein binding a single light harvesting pigment, and another very big multi-subunit protein called the phycobilisome, which binds almost 400 pigments. When OCP, which is a photoactive protein, when it is illuminated with blue light, it changes its structure, which allows it to bind to this blue subunit of the phycobilisome. And upon binding, the measured fluorescence intensity and lifetime of the fluorescence suddenly decreased, which means that the photosynthetic activity of the phycobilisome was switched off. So we illuminated OCP with blue light, and then after a few seconds it bound to the core of the phycobilisome and switched off its photosynthetic activity completely. In the bottom example, it is shown that this behavior can be reversed. After some time, the intensity and lifetime recovered, which means that the OCP detached again from the phycobilisome core. Now, if you look carefully here, you will notice that there are very short intermediate states in this switching off and switching on. And we have analyzed these intermediate states and have identified an interesting docking mechanism that takes place during the binding process of OCP to the phycobilisome core. Interestingly, when you shine light on such a phycobilisome, the emission intensity jumps between discrete quasi-stable levels. These large intensity jumps indicate that the photosynthetic activity of this system is basically switched on and off the whole time. It's pretty amazing that such a large system, which is basically a network of almost 400 pigments, that this large system can make such large changes in the emission intensity. This means that this is a diffusion limited system and that these low intensity states correspond to energy traps that effectively trap every long living excitation. Let's now add the lifetime resolved for each of these intensity levels. Basically, all the intensity and lifetime switches are linearly correlated. And this plot shows us two things. Firstly, the slope of the fit tells us how large the phycobilisome complexes are, ensuring that we worked only with intact complexes, because they fall apart quite easily in this environment. Smaller systems will make this slope steeper. And secondly, the linear correlation tells us that energy quenching occurs mostly at the terminal emitter, because this is a diffusion limited system. If there is a quencher before the terminal emitter, where a fraction of the energy gets lost, this would pull the correlation non-linearly upwards, because the lifetime is still determined by the terminal emitter, so only the intensity decreases. We studied the fluorescence intensity switches across a broad range of excitation intensities. Shown here on the left is an example of a trace at low light and one at high light intensities, indicating a huge difference in the switching frequency. Let's use a simple two-state model to parameterize the switches. We define a switch, an intensity decrease from a bright to a dark state, or an unquenched to a quenched state, and an intensity increase we define as a switch from a quenched state to an unquenched state. Now, plotting the switching frequencies of both switches as a function of the excitation intensity, we notice that the frequency of both switches increases significantly with the light intensity, which means that both of them are light induced. Just going back there. So we went down to extremely low excitation intensities in the context of single molecule experiments, corresponding to physiologically relevant light intensities. And it's the first time that such low intensities were used in a single molecule study. When you look carefully at this plot, you'll notice that the black curve is slightly steeper than the red one. So when I take the ratio between the two, this ratio also increases with enhanced illumination, indicating that the net effect is an equilibrium shift towards quench states, exactly what one expects from a photoprotective mechanism. Because when there's more light, more photo damage can take place, so more of the excitations must be quenched in order to become harmless. And this quenching is just thermal dissipation, harmless, getting rid of the excess energy in a harmless way. So this is a new intrinsically available photoprotective mechanism that is switched on immediately and with greater effect as the excitation intensity increases. To determine the possible involvement of the protein scaffold in the intensity switches, we made use of a chemical cross-linker, which binds to the proteins and makes them stiffer. It's like putting a soft brace on your knee. So you're not going to prevent all the motion, but it's going to be stiffer. Now, as the amount of cross-linker was increased, in this case from 0 to 1%, the switching frequency decreased significantly for both types of switches, which gives strong evidence that the intensity switches are governed by protein conformational dynamics. Now, to determine the underlying molecular mechanism, we can do some modeling and simultaneously measure the spectra. I used a compartmental coarse-grain model based on time-resolved emission data and Boltzmann statistics to show that quenching takes place most likely in the core. So these black and blue subunits, they represent the core. And the reason for this is that the experimental data correspond mostly with the blue and the red curves here, which indicate that the quencher most likely acts on this black compartment and sometimes also the blue compartment here. Now, by simultaneously measuring the fluorescence spectra, we observed a fascinating phenomenon, and that is that the phycobilisome complexes occasionally displayed enhanced red emission, shown in the spectrum here. So besides the normal emission peaking at around 670 nanometers, it also occasionally exhibited emission in the far red. And in 80% of the cases, this red emission was preceded by a long-living quench state, a low-intensity state, which suggests that the quench state must be associated with the red state. It suggests a relationship between these two just because of the very high prevalence. Further investigation indicated that both the quenching and the red emission originate from a charge transfer state that is formed in one of the light-absorbing pigments. So we managed to resolve the molecular mechanism that's involved with quenching in phycobilisomes. The quenching site is mostly one of the core subunits of phycobilisome, which serve as the terminal emitter site. And the mechanism is that light induces a protein conformational change, which in turn induces a pigment configurational change, forming a charge transfer state, which is the dark state that traps the energy. In the last example, I'd like to show how biomolecules can be interfaced with nanoparticles to actively tune their radiative and non-radiative rates. We will deal with localized surface plasmons. When the dimensions of a metallic particle are smaller than the wavelength of the incident light, the conduction electrons on the surface oscillate collectively in resonance with the frequency of the incident light. And this is known as the localized surface plasmon resonance. Now, if you take a gold nanorod, or nanoparticle, but nanorod in this case, and excite it with laser light, the radiation field from the laser interacts with the surface plasmons, and the result is localization or concentration of the electromagnetic energy in hot spots close to the surface of the nanoparticle. Now, as you bring an emitter, a molecule or a light harvesting protein in our case, if you bring this emitter closer to the metallic surface, the fluorescence intensity will dramatically increase until the emitter gets too close to the metallic surface, in which case quenching will quickly become the dominating process. Metallic nanorods have two surface plasmon modes, one along the long axis, called the longitudinal mode, and one along the short axis, called the transverse mode. When you change the length of the nanorod, the absorption and emission spectra can be shifted quite substantially. In one of our recent projects, we have chemically synthesized gold nanorods and spin-coated them on a glass substrate. And then on top of this, LHC2, the main light harvesting complex of plants, was spin-coated. The dimensions of the gold nanorods were such that the absorption band of the longitudinal plasmon mode overlapped strongly with the absorption and emission bands of LHC2 to ensure optimal interaction between them. We then investigated the distance-dependent interactions between a single nanorod and a single LHC2 complex. So when a single protein is in or near the plasmonic hotspot at the tip of a gold nanorod, the fluorescence or photoluminescence intensity got enhanced by as much as 2 orders of magnitude. Compare the intensities here of a single LHC2 complex far from a nanoparticle with this strongly interacting hybrid system, showing a 2 orders of magnitude difference between the fluorescence intensity that was measured. This photoluminescence enhancement is accompanied by a significant reduction in the lifetime, the fluorescence lifetime. In other words, both the radiative and non-radiative rates are drastically tuned due to this interaction between the emitter and the plasmon modes. The fluorescence enhancement is a product of excitation enhancement and emission enhancement, parameters that depend on factors like the nanoparticle size and its distance from LHC2, and all these factors can be precisely controlled. So one can actually get a handle on the radiative and the non-radiative rates of this hybrid system. This can all be modeled in a relatively simple manner. One needs to add three terms to the excitation-de-excitation cycle. The first is the excitation rate enhancement. The second is the Purcell factor, which describes the emission rate enhancement. And the last is an additional decay channel, de-excitation channel, which indicates energy transfer from the emitter to the metallic nanoparticle. And combining all of this, one can calculate the fluorescence quantum yield of the emitter in terms of these rates and its intrinsic quantum yield, Y naught. Okay, so with this, I would like to conclude and shown here is an earlier version of my research group around the single molecule spectroscopy setup at the University of Pretoria and all the parties that were responsible for funding to make different parts of this research possible. And please feel free to contact me at any time to ask questions. I'm always happy to answer questions. Thank you very much.